All right, now let's finish up chapter 14, the third section. So we talked about the fact that we can sequence the DNA of a whole chromosome or parts of a chromosome, whatever. Well, back in the 1990s, the early 1990s, they started what became known as the Human Genome Project, and the goal was to sequence the complete genome for people, okay, for humans. And so um, early on, this process was rather time-consuming and laborious because doing the sequencing took a lot of people to do it and time and all that kind of stuff. Um, but they were able to develop techniques that sped the process up, and so the human genome was completed sooner than they thought. Um, Now, why would we want to sequence the complete human genome? Well, as we say here, to explore gene function. So essentially to find genes and hopefully to figure out what they do. Um, and to study the variation that exists within and between people. Um, so we did find that, well, okay, let's talk about that advanced technique first. So what developed, what became known, and this is mentioned in your book, it's known as the shotgun technique. And what you do is you take one big piece of DNA, say a whole from a whole chromosome, and you cut it up into pieces. All right. You separate those pieces, and then you sequence the individual pieces. All right. And you'll cut that DNA with a couple different types of restriction enzymes. So what you have end up with are pieces that overlap each other. Okay. And where the overlaps are, you get a match. And so you sequence smaller pieces, which makes the job easier. But then you can use computing power to basically match up all those pieces and figure out the sequence for the large piece of DNA. Um, Now, once you've got your sequence for a complete chromosome, say, or the whole genome for that matter, how do you find where the genes are? Well, there's a couple different things you can look for. We talked earlier about how genes have a promoter before them, and the promoter, you'll recall, is where transcription factors go um, when you want to transcribe the gene. And again, the transcription factors help the RNA polymerase to find where the gene is. So you can look for promoters. Um, you can also look for start and stop codons. Okay, so you can look for promoters and start and stop codons. And so where you find start and stop codons are pretty good indicators of where you have genes, because you remember we said that a gene will always start with the codon for methionine, and there are those three different types of stop codons. Now, you can sometimes find methionine codons in between because it can occur in other places, but it'll always be at the very beginning. Um, and so once you've got that sequence, once you start finding genes, um, again, we can compare different people. And in the initial project, they didn't sequence the genome of just one person, but they sequenced multiple people. And now more and more people's genomes are being sequenced, and other species are being sequenced. And so a lot of information is being gathered and a lot of knowledge gained. And of course, when you look at, as I think I mentioned earlier, the genome of any two people, they tend to be quite similar because, of course, we are all the same species. But as you can see, on average, about 1 out of every 1,200 bases does not match. And when you get these single bases that don't match up between people, or they tend to vary between people, these are what are called SNPs, or single nucleotide polymorphisms, and these are found throughout the genome. So in these three people at this particular portion of their genome, it's very similar, but there just happens to be this one base that has changed. And so these would be due to mutations that have occurred over time and been passed down and cause the variation between people. Now, the field that has grown significantly with the advent of DNA sequencing technology and the generation of the human genome is what's called bioinformatics. And that is, it's combining basically computing power with biology and taking all of that sequence data, all that genetic information you get from different species, um, plugging it all into computers and using computing power to try to make sense of it, to find those genes, to study the variation that occurs between individuals, between species. It would be very hard to do this if you just had to look at sequences on pieces of paper. And so the fact that we're able to use computing power nowadays has really helps enhance our knowledge and speed up the process. And so essentially using bioinformatics, we're studying what you would call genomics, when we study genes, or genomes, genes, and their functions. And this is a very big field within biology these days. All right, so throughout the 90s, the Human Genome Project was occurring. In 2000, so after it took about 10 years, which was less time than they thought it would take, um, the human genome was basically figured out, was deciphered. And so if you look at our haploid genome, that is just one side of the DNA chromosome. The whole genome is about 3 billion bases long. That's pretty darn long. Again, that's comprising all of the chromosomes we have. Might be surprised that only about 2% actually codes for proteins. So there's a lot of extra DNA. So within any given chromosome, there tends to be a lot of sequences in there that don't contain genes. All right. So on a given chromosome, you might have a gene here, a gene here, a gene here, but you can have large gaps in between where there's no gene, no actual gene there. By comparing our genomes to other organisms, we have found that a good chunk of our genome is derived from viruses and other sources. This might be surprising, but of course viruses have been around as long as other living things have been around, and so viruses sort of survive by and reproduce by injecting their DNA into some host organism, into the cells of that host, and basically over evolutionary time, that virus DNA can become incorporated and become part of the genome of that host organism. If you have ever, maybe you guys haven't because of the vaccine, but if you've ever had chicken pox, the chickenpox virus basically leaves its DNA inside of you, inside some of your cells, and it basically becomes a part of you. So also when we compare other species, we see that there's a fair amount of similarity. There are certain genes that basically all living things share. And so it's estimated that up to 40% of our proteins and essentially 40% of our genes are similar, whether you're a person, a fruit fly, a worm, yeast, whatever. I think I gave the example in class maybe of, when you think of the process of cellular respiration, this is something that just about all organisms do, and so there are certain proteins needed to do this, which means there are certain genes needed to make these proteins, and so those will be genes that will basically all share. Now, if you looked at those, if you compared those genes between a fruit fly and a person, the gene would be the same, but of course there would be some variation within that gene to a certain extent. So, of course, we're also using this to identify genetic diseases. I've identified approximately 25,000 genes in the human genome. The function of many of them has been found, but there are still many more that we know there's a gene in a particular portion of the chromosome or of the genome, but we haven't necessarily figured out what that gene is doing, and so that's another big area of research and exploration. So again, other species, their genomes have been sequenced, and here are just a few examples. So you can see ours was a little over 3 billion bases long, about 25,000 genes. You can see that mice, about 2.5 billion bases, 24,000 genes more or less. And so you can see some of the variation in genome size and the number of genes. And viruses have very small genomes and relatively few genes compared to living things. Viruses, by definition, really aren't alive because they don't have a metabolism and they can't reproduce on their own. But we, of course, study viruses in biology because they affect living things to significant degrees. A lot of these organisms on this list, these non-human ones, are basically organisms that have been studied extensively for various reasons. So they were some of the first whose genomes have been sequenced. Not on here, but we might take a look in class and compare. Of course, the chimpanzee is, as far as we can tell, our closest relative out there in the animal kingdom, and so the genomes of humans and chimps are quite similar because of our shared recent ancestry. All right, that's it for chapter 14.